Let's go ahead and take a look at the events that happen at the neuromuscular junction. So we know that skeletal muscle is controlled by a somatic motor neuron, right? So there's a synapse between a somatic motor neuron and the muscle fiber, right? So this is called the neuromuscular junction, where the motor neuron serves as the presynaptic cell and the muscle cell, the myofiber, serves as the postsynaptic cell. So let's just go ahead and draw sort of this synapse here, right? So here is our motor neuron, okay? And remember, our motor neurons, they're myelinated, so we have sort of this cell right here, right? And then we've got, let's put one over here too, like this, right? These is the myelin sheath. So I'm going to just sort of label that there. It's myelin. This is myelin here. Okay, and then down here, we're going to have our muscle fiber or myofiber, right? And so the portion of the myofiber that is part of the neuromuscular junction, right? So the folded area, that's known as the motor end plate, right? So this is your motor end plate. I'll abbreviate MEP, motor end plate. All right, now, a couple things that we have to put in place here. The first thing that we have to put in place is we have to put sort of these vesicles in the axon terminal or the synaptic bulb that are going to contain a neurotransmitter. And we know that since this is a somatic neuron, that neurotransmitter, right, is going to be acetylcholine, right, or ACH. Okay. The other thing that we have to put on our cell is we have to put our voltage gated calcium channels, right? So I'll label that sort of up here. This is our voltage gated calcium channel. Then on our motor end plate, we are going to have, we'll draw these in red, these are going to be ligand gated sodium channels. And the reason that that motor end plate has this highly folded sarcolemma or plasma membrane, right, is so that we can have an increase in concentration of those ligand gated channels. Okay. Now, on the periphery of the motor end plate, we'll draw these in yellowish, right? Over here, this sort of ridge here, this is known as Doyer's eminence, right? And on Doyer's eminence is where you are going to see some voltage-gated sodium channels. So we're going to put, let's go ahead and draw our red ones here. This is going to be our ligand-gated sodium channel. And then in our yellowish color, right, this is going to be our voltage gated channel. And this is a sodium channel as well. So Na+. All right, so now what's going to happen? So remember that in the motor neuron, when we generate the action potential, right, the action potential propagates down the motor neuron. And because this is a myelinated neuron, it's going to use saltatory conduction. So it's going to, we're going to say the action potential reaches the axon terminal. So that's where we're going to start. So if you don't recall how action potentials are generated in neurons or how they propagate down a neuron, make sure that you go back and you watch those videos. Okay, so the next thing that's going to happen is you're going to get an activation of those voltage gated calcium channels. So we're going to say voltage gated calcium channels open. And so when those voltage gated calcium channels open, that calcium is going to influx into the cell. So step 3 is we get a calcium influx. And remember that this calcium is going to come over and it's going to bind to those acetylcholine containing vesicles. So we're going to say calcium binds to the acetylcholine containing vesicles. All right. So when that happens, these vesicles are going to come over and they're going to fuse with that neurolemma, that plasma membrane of the motor neuron. And so when they fuse with the neurolemma, right, so that's going to cause exocytosis. So we're going to say vesicles then fuse with the neurolemma, which causes exocytosis of that acetylcholine. So the acetylcholine then ends up in this space, right? So acetylcholine ends up into the synaptic cleft. Remember, that's what that space is called, the synaptic cleft. Okay? So when acetylcholine gets moved into that synaptic cleft, it's going to come over and it's going to bind to that ligand-gated sodium channel. So step 7 here, we're going to put our ACH binds to ligand-gated sodium channel. Okay. I'm going to just move this up just a little bit here so we can get a little bit more room to write. Right? And when that channel opens up, right, so the ligand's going to bind, the channel is going to open up, and that's going to cause sodium influx into the myofiber. So we're going to sort of put that as one big step here. So we're going to say the ligand-gated sodium channel opens, which is going to lead to an influx of sodium. Okay, so now we have this sodium inside of the myofiber at the motor end plate, and it's going to diffuse away from the site of entry, right? So it's going to create a graded potential at the motor end plate. So we'll say graded potential at motor end plate, right? And so if you don't remember how graded potentials work, remember to go back and watch those videos on graded potentials in neurons. But you're going to create a graded potential such that threshold is reached at the periphery of the motor end plate. So I'm going to just move this up again. Okay, so step 10 is we're going to say threshold reached at periphery of motor end plate. Remember that periphery where it's raised is called Doyer's eminence, right? So periphery of motor end plate. And when that happens, we have these voltage-gated sodium channels. So the activation gate, remember voltage-gated sodium channels have that 2 doors, right? So our activation gate on the voltage-gated sodium channel opens, right? So what's going to happen then is you're going to get a bigger influx of sodium into that cell. So we'll say influx of sodium, and that's going to cause that membrane potential to rise, which is going to lead to an action potential in the myofiber. Okay, so all the events that sort of happen at the neuromuscular junction, right, all right here. So what you have is you have this action potential coming down, right? Comes down the motor neuron, activates the voltage-gated calcium channels. Calcium influxes into the cell. Calcium is going to bind to the vesicles, which are going to cause them to fuse with the neurolemma, exocytosing the acetylcholine into the synaptic cleft. Acetylcholine will bind to ligand-gated sodium channels on the motor end plate, which will cause them to open up, allowing sodium to influx into the myofiber or the muscle cell. That will create a graded potential, and threshold will be reached at the periphery of the motor end plate, activating the voltage-gated sodium channels, which will cause the activation gate on the voltage-gated sodium channels to open up. This allows sodium to influx into the cell. We get this major influx of sodium into the cell, and that generates an action potential in the myofiber at the periphery of the motor end plate. So those are the events at the neuromuscular junction.